The human brain is the most complex living structure. The brain of an adult human contains billions of cells which are called neurons and glia. It is the communications between neurons which enable people to think, move, and feel emotions. Normal function of the brain is dependent upon the adequate supply of oxygen and nutrients supplied through a dense network of blood vessels. The location of the damaged area of the brain will lead to variations in specific neurological deficits such as impaired speech, vision, or movement. The stroke mechanisms animated here reveal the involvement of the NMDA receptor in the early diagnosis of brain damage. Stimulated neurons use electrical impulses to relay information along the axon and to other neuron cells. There are two categories of strokes, those caused by a blockage of blood flow due to the formation of a clot within a cerebral blood vessel, and those caused by bleeding. The clot restricts oxygen and glucose supply to the neurons. This results in an electrical signal interruption which leads to neuronal death. In this cross-section, the microvessel is enclosed in glial cells, green, and axons, which are in intimate contact with microvessels. Nervous system capillaries are exchanging nutrients, glucose, and oxygen with brain cells through the cytoplasm and endothelial cell that makes up the capillary, red cells. Endothelial cells form the blood-brain barrier which protects the neurons and glial cells. As a result of blood flow restriction, the blood-brain barrier is compromised. Neurons affected downstream of the blockage release excessive glutamate, a major excitatory neurotransmitter. Glutamate binds to NMDA receptors, triggering excessive influx of calcium and the receptor's overstimulation and overexpression. Overwhelmed and unregulated, the activation of NMDA receptors allows for unchecked entry of calcium, which compromises the ability of neurons to recover from osmotic stress. After an ischemic event, calcium influx initiates thrombin-activated serine proteases, cleaving extrasynaptic N-terminal domains from NR2 subunits of NMDA receptors. This releasing of peptide fragments causes receptor degradation. In these conditions, the neuron cannot recover and begins shutting down. Peptide fragments move through the compromised blood-brain barrier into the bloodstream. The immune system recognizes brain peptides as foreign antigens and generates antibodies. It was demonstrated that NR2 peptides and antibody amounts correlate with ischemic areas. The detection of these biomarkers in blood may improve diagnostic certainty of cerebral ischemia and will be beneficial for stroke management. Better understanding of the stroke mechanisms will improve diagnostic certainty in emergency situations and might predict the risk of silent brain damage before an acute cerebrovascular accident, benefiting the patient as well as the healthcare system.